Okay, great. This is Cynthia Powell from UNC Chapel Hill, and I'd like to welcome everyone to the webinar about the North Carolina newborn screening pilot for mucopolysaccharidosis type 1 or MPS I, what healthcare providers need to know. Before we get started, I'd like to acknowledge the National MPS Society for supporting this webinar and also the website connected with this. Next slide. I'll go over the agenda for today. Next slide. I'm Cindy Powell. I'm a clinical geneticist at UNC Chapel Hill in the Division of Pediatric Genetics and Metabolism. And I'd like to introduce the other speakers and give a brief overview about conditions that are screened for in newborns. Next slide. Dr. Joseph Munzer is Professor of Pediatrics and Genetics here at UNC Chapel Hill. He's a biochemical geneticist, and he's considered one of the world's experts in the mucopolysaccharidoses. And he'll be speaking about the clinical features and treatment for MPS I. Next slide. Dr. Jennifer Taylor is a chemist, and she did postdoctoral work at the Centers for Disease Control and Prevention in Atlanta in newborn screening, and she's a senior research analyst at RTI International. She'll be talking about the laboratory part of newborn screening for MPS I. Next slide. Ms. Kristen Kleinard is a genetic counselor with us in the Division of Pediatric Genetics and Metabolism. She is our primary genetic counselor working with our metabolic patients, and she'll be talking about the clinical follow-up for newborns who screen positive for MPS I. Next slide. I'd also like to acknowledge other members of our leadership team for this pilot. At RTI, Dr. Donald Bailey, who heads up the team, Dr. Lisa Getland, also at RTI International, Dr. Alex Kemper at Duke, Dr. Scott Zimmerman, who's the director of the North Carolina State Lab of Public Health, Mr. Harry Patel, who's in charge of the Tandem Mass Spec Newborn Screening Laboratory, and Dr. David Millington, retired professor from Duke University, who's one of the world experts in use of tandem mass spec in newborn screening. Next slide. I'd also like to thank our advisory committee members, including Dr. Rusher, Ms. Wiedehase, Dr. Zolatar, and Mr. Dant, and the laboratory directors at Duke, Dr. Bally and Dr. Rader. Next slide. So to give a little bit of an overview about newborn screening conditions and how conditions are chosen to be screened for in newborns. Next slide. So in order to have more uniformity across the United States in terms of what conditions various states are screening for, in 2003, a committee, now known as the Advisory Committee on Heritable Disorders in Newborns and Children, was begun by the U.S. Department of Health and Human Services, made up of experts in pediatrics and newborn screening. They determine conditions that are part of what's called the RUSP, or the Recommended Uniform Screening Panel. Conditions can be nominated for the RUSP, and then a systematic evidence-based review is done. Next slide. This evidence-based review looks at the potential benefit of doing newborn screening for the condition, and also looks at capabilities of state labs to perform the screening test. Next slide. The committee then votes on the condition in question, and if the committee recommends that a condition be added to the RUSP, this then goes to the Secretary of Health and Human Services for final approval. Now, even if a condition is added to the RUSP, it's still up to individual states to decide whether to add it to their newborn screening panel. And states are also able to add conditions that have not yet been approved for the RUSP. In North Carolina, adding a condition to the newborn screening panel requires legislative action, approval of a bill by both houses, and the governor's signature. Next slide. This is a list of the current 34 core conditions on the recommended uniform screening panel. And next, I bring your attention to the bottom of that slide. Next, click again, please. To MPS I or mucopolysaccharidosis type 1. Click. This was added in February of 2016. To give you a little bit of a reference point, I'll point out the one just above that, SCID, or severe combined immunodeficiencies. Click again, please. This was added in May of 2010. Approximately half the states in the U.S. are screening for SCID at the present time, and North Carolina hopes to add this to their screening panel later this year. Next slide. Currently, 4 states in the U.S. are screening for MPS I. To assist in the implementation of newborn screening for MPS I, the National Institutes of Health, through the National Institute of Child Health and Development, issued a task order to fund pilot studies in states to do this. Contracts were awarded to North Carolina with involvement of RTI, the State Newborn Screening Lab, UNC, and Duke, and also to the state of Georgia with their State Newborn Screening Lab and Emory. And so it's this pilot study that we're referring to today. Next slide. I'll now hand it over to Dr. Munzer to talk about the clinical features and treatment of MPS I.
Thank you, Cindy. It's certainly my pleasure to be involved in this exciting newborn screening project, and also my goal now is the next few minutes present some of the clinical features and the treatment options for patients with MPS I. Next slide, please. MPS I is one of a group of disorders called mucopolysaccharidoses, or obviously use MPS for short. These are a series of lysosomal enzyme deficiencies. We now know of 11 known enzyme deficiencies that comprise 7 different clinical types. Each individual disorder is quite rare, but as an aggregate, we estimate there's 1 in 25,000 patients are born with an MPS across the U.S. The hallmark of these diseases are the tissue storage of glycosaminoglycans. These are complex carbohydrates that need to be recycled. When they're not recycled, they get stored and cause problems we'll talk about. These are progressive disorders due to this ongoing storage, which results in multi-systemic involvement across the different disorders. As we see for most genetic disorders involving enzyme deficiency, there's a wide range of clinical involvement in each of the MPS disorders. Next slide. Here's an example of a patient with MPS I, a 4-year-old. MPS I is due to the deficiency of the lysosomal enzyme alpha iduronidase. Next slide. The biochemistry of the disorder is when you're missing iduronidase, this results in a block in the sequential breakdown in the glycosaminoglycans. Normally what happens is that you need a series of enzymes to cleave different bonds, and only the non-reducing terminal end is potentially cleavable. And so when you're missing one enzyme, the whole process stops. Next slide, please. This shows you an example of not sure if you can, there's a 2 slides superimposed. I'm not sure what's happening, but you can see Thank you. On the right side is a fibroblast from an MPS patient, and all the clear vacuoles are very enlarged lysosomes that have been accumulating, glycosamine glycan, and this results in both cell dysfunction and some cell death. Next slide, please. Next slide. So again, in clinical features, the onset of symptoms in the severe form of go back one, please. The onset of clinical features in the severe form of MPS I typically occur before 6 months of age, and we refer to the severe form as Hurler syndrome. Historically, Hurler syndrome used to equal all of MPS I, but now we reserve Hurler syndrome for just this severe form, as I'll discuss with both physical and cognitive involvements. The severe form, children typically untreated die between 3 and 10 years of age, and we recognize this to be a rare disorder with an estimated incidence of 1 in 100,000. Next slide, please. As we talked about, there's a spectrum of involvement in MPS I, and we already alluded to Hurler syndrome, the severe form with severe cognitive impairment. They have upper airway disease, respiratory disease, cardiac disease in terms of valve involvement, hearing problems, and if untreated, as I alluded to, they die before age 10, some as early as 3 or 4. At the other end of the spectrum, we refer to attenuated patients, or Shea syndrome, which are normal intellect, have less progression of their physical disease. They have corneal clouding, as does Hurler, joint stiffness, valve disease, but typically can live into adulthood, and some even live a normal lifespan. And we have an intermediate group historically called Hurler-Shea that have combinations of some involvement of the Hurler and Shea, but it's really more intermediate. But it's very difficult to place any given patient along this spectrum since it's a continual spectrum, and it's sort of artificially call it 3 forms, but that's historically what has been done. Next slide, please. Here's an example of 3 individuals with MPS I. Patient with Hurler syndrome, Hurler-Shea on the far right, a Shea patient. All these patients have virtually undetectable enzyme activity. The Hurler patient absolutely has no detectable enzyme activity, and the Shea patient may only have half of 1% true residual activity, which is enough to spare the brain but still cause physical disease. Next slide. As we alluded to, this is a progressive disorder, and you can see the picture on the upper left, the 10-month-old. If you just look at her face, she doesn't look like she has a storage condition. Go back one slide, please. But if you examine the rest of her, since I've known this child, you would recognize she had evidence of storage in terms of joint involvement, hepatomegaly. But clearly at the lower right, at 39 months of age, it's obvious a child with facial features of a storage disease, just showing how the disease progresses. This is basically a child untreated. She'd received enzyme therapy, but after the 39 months. Next slide, please. So there is a genotype-phenotype correlation in MPS I. In this disorder, we make the statement that a nonsense mutation, that is a mutation that results in a premature stop, results in total lack of enzyme activity from that particular allele. We're aware in MPS I that if you're homozygous for a nonsense mutation or a compound heterozygote, that is 2 different nonsense mutations, that's always severe disease. And we now know at least 15 known nonsense mutations, with 2 being relatively common, of Northern European ancestry, W402X and Q70X. And so there's a high likelihood that we can predict the phenotype based on genotype in many of the MPS patients because of this prevalence of the nonsense mutations. We previously knew that there were pseudodeficiency alleles, either alleles that result in decreased enzyme activity but not enough to result in any clinical disease. And with newborn screening, we're recognizing these are much more common than we previously thought. Next slide, please. So what are the current treatment options for MPS patients? In general, we have 2 treatment options. Historically, hematopoietic stem cell transplantation, initially with bone marrow donor and now with cord blood. And then more recently, intravenous enzyme replacement therapy. Next slide, please. This slide shows an overview of the treatment options for patients with MPS based on their diagnosis, and then in terms of whether hematopoietic stem cell transplantation or ERT. You can see on the right, ERT is available for 4 different MPSs currently. Hematopoietic stem cell transplantation is beneficial, for the physical disease in MPS I and MPS VI. Next slide. Just a highlight showing you that in MPS I, we basically transplantation. It's done young, as I'll show you in a second, and a Hurler patient can actually prevent the neurologic deterioration that occurs in this disorder, where the other forms of MPS, hematopoietic stem cell transplantation, have not been proven to alter the brain progression that occurs. Next slide. This is a patient of mine who's the first patient with MPS I who has ever received enzyme therapy. It's a 16-year-old, 17-year-old with Hurler-Shea syndrome. If you saw him, he has coarse facial features, he has corneal clouding, he has a tracheostomy, he has hepatomegaly, umbilical hernia. This is the best he can stand because of stiff joints. After 6 months, he had improvements, but this is the best he ever got. I use this slide to emphasize that it's much better to try to prevent these physical features than trying to correct them. And that's why newborn screening is so important, to identify patients early for transplantation and also identify the attenuated patients prior to developing significant disease to try to prevent that. This individual was intellectually intact. Next slide. So here's just the schematic before newborn screening of how we dealt with patients who were detected with MPS I based on clinical features. And we really separated whether they were younger than 2 years of age or 2 years of age and older. And if you had a mutation that predicted severe or unknown phenotype, and you had a normal cognitive ability, which most of the young Hurler patients do, then transplantation is really the treatment of choice. And today, transplantation is the treatment of choice for the patient with MPS I, who has a Hurler syndrome under 2 years of age. And currently, most everybody has a donor since the source of the donor is cord blood. Patients who are greater than 2 years of age typically recommend enzyme replacement therapy, and only in very rare occasions do we recommend a transplantation for those patients. Next slide. That should be the end of my presentation. Thank you.
Thank you. Now we'll go on to Dr. Taylor.
Hello. Can everybody hear me?
Yes.
Yes. Thank you. All right. Thank you, Dr. Powell, for letting me talk, and I am also very excited to participate in this pilot study. And we're going to switch gears a bit, and we're going to talk about what's happening in the laboratory, how the screening test works, and how it will work here in North Carolina. Next slide. So first, I'm going to give you an overview of what we're doing here. So we are going to, in this pilot study, as Dr. Munzer stated, the enzyme that is deficient is the IDUA enzyme. So the first-year screening test that we're going to be using is looking at the functionality of this enzyme. We are going to be screening from the existing blood spots that are sent to the North Carolina State Laboratory of Public Health, and the testing will take place in these facilities. How this test works is it is an enzyme activity assay. So what we will do is we will take the dried blood spot specimen and incubate it with a synthetic substrate and internal standard. If that specimen is normal, then the IDUA enzyme will cleave the substrate to produce a product. And this product can be detected using tandem mass spectrometry. Now, in other newborn screening tests, the mass spectrometry results, they usually report out a concentration. Well, since this is an enzyme activity assay, the results are a little bit different. We're looking at the concentration of the product over time, which is enzyme activity. So the results that are calculated are micromole per liter per hour. And what to expect is in normal newborns, the activity is very high, so you'll have a high result. But in the screen-positive infants, you have no or very low activity. So if you have a screen-positive result, then that sample will then be sent for molecular testing. The IDUA gene that produces the enzyme will be sequenced at the Duke Molecular Diagnostic Laboratory. Next slide. So this is an overview of how we process a sample for the first tier test in the state lab. What we do is we punch a 3.2 millimeter DBS punch into a 96-well plate, and each well in that plate is incubated with 30 microliters of cocktail solution. And in this cocktail solution, you have the substrate and the internal standard. So that is incubated overnight, and after 19 hours, it is then quenched with a solution composed of methanol and ethyl acetate. This solution deactivates the enzyme so it does not cleave the substrate anymore. Then part of that quenched solution is then transferred into a deep well 96-well plate where a liquid-liquid extraction occurs. You add ethyl acetate and water, and if you let that solution sit, it separates in 2 layers. And the top layer is then transferred to another 96-well plate, dried using a liquid, nitrogen dryer. And then it's reconstituted using a reconstitution solution made of ammonium formate and methanol. And that solution is shaken for a bit and injected in the mass spectrometer to analyze the product that the enzyme produces. Next slide. So now I'm going to go over how the screening algorithm will work for the state lab. So again, we're testing for the IDUA activity, so we'll do an initial test with 1 punch. If that activity is above our cutoff, then no further testing will take place. As you guys can see, if you follow along from the upper left-hand side going across. If it is below the cutoff, then we will retest that sample in duplicate. And then we will average those 3 results, so the initial and the 2 results from the retest plate. And if that is below reporting cutoff, if that's above a reporting cutoff, then no further testing will take place. However, if it is below this cutoff, then that sample will be sent for DNA sequencing. Now, if you look on the lower right-hand corner, from the DNA sequencing, if you have 1 or more mutations identified, so those are the mutations that Dr. Munzer talked about in his presentation. Those are variants of clinical significance, variants of unknown significance, and also these pseudodeficiency variants. Those will be considered screen positive, and those infants will be referred to follow-up. If you look on the lower left-hand side, if there's no mutations that are identified from DNA sequencing, then a repeat newborn screening specimen will be requested, and then that specimen will go through our algorithm again. If that second specimen is above the cutoff, then no further testing is needed. But again, if that second specimen is below the cutoff, that will also be referred to follow-up sample. Next slide. So a little bit more about our pilot study. This is a statewide pilot study, and we're screening 80,000 infants. So all specimens that come into the state laboratory will be screened until we hit that number of 80,000. The pilot will begin in August 8th, and is projected to last till May of 2017 until we hit that 80,000 infant number. Some important things to know is the samples will be tested for this pilot 3 days after it is received in the laboratory. So the reason why is the sample will be tested by the standard newborn screening test first, and then after those tests, it's been tested through that, then they will be tested for this test afterwards. And results will not be reported on the newborn screening report since it's 3 days later. If you have a positive result, you'll be contacted by the genetic counselor, which we'll discuss next. But one thing I did want to notice that you might also be contacted if there's an inconclusive result and we need a new specimen to be collected and sent to the laboratory. Thank you. Next slide.
Thank you. Now, Ms. Kleinert will talk about the follow-up protocol in clinic.
Thank you. So again, I'm very excited to be included on this project as well. Can everyone hear me okay?
Yes.
Okay. So lastly, I'll be talking about the follow-up for those who screen positive on the newborn screening for MPS I. Next slide. So we've mapped out a flow chart that will outline the step-by-step process that we plan to follow with the abnormal or positive results. So once the state lab has identified an abnormal screening result, as mentioned, they will contact us at UNC either by phone or fax to notify us of the abnormal screen. Once we've received that result, we will then make a call to the primary care physician to notify them of the screen results. Then either the primary care physician or the genetic counselor myself will contact the family directly in order to schedule an appointment with the newborn and parents here with us at Chapel Hill. Next slide. So now the family's been contacted about the abnormal screen. An appointment at UNC has been scheduled. So the day of the appointment, the family comes to UNC to be seen by the geneticist as well as genetic counselor. At that time, we will obtain samples for confirmatory testing for MPS I, which will include a urine sample for urine gags and also a blood sample for enzyme testing. These samples will be sent to Duke, who will then notify us of the results. If both the urine gags and enzyme levels are negative or normal, then the conclusion is that the newborn screen was likely a false positive and no follow-up is needed. If the urine gags are negative or normal and the enzyme level is decreased, then the conclusion is that this is likely a pseudodeficiency. But we would like to have the child and family back for a 12-month follow-up. If the urine gags are positive and there is also a decreased or absent enzyme activity, then these results do confirm the diagnosis for MPS I. And as we mentioned with the mutations, so if there are the nonsense mutations in the IDUA gene then that are associated with the severe form of MPS as well. If these mutations are identified through testing, then we will refer the family and child to Duke for the stem cell transplant. If these mutations are not identified through testing, then the child will be followed every 3 months at UNC for evaluation and to determine the need for the enzyme replacement and or stem cell transplant. Next slide. So just to kind of wrap everything up, if you receive a call from UNC informing that the patient does have a positive newborn screen for MPS, we will inform the parents that this is, again, a screening test, and we will need to have confirmatory testing that we discussed with the urine and blood sample that will all be arranged at UNC once we've scheduled that appointment. Just to also note, we will not know the rate of the false positive screens because until we actually get the data from the pilot study. And then also, if the family or infant is found to have MPS I, there are treatment options, as Dr. Munzer had discussed in his talk as well, that are available and will be reviewed with the family. Next slide.
So that concludes the formal part of this webinar. The webinar will be posted, and there's additional information about the pilot at the website that you see the URL there. And at this time, we'd like to take any questions that anyone may have. All right. Hearing none, we'll conclude the webinar. Thank you very much for participating.